So let's go ahead and talk a little bit about CRISPR. So what CRISPR is going to do, I'm going to rely mostly on this slide right here because it's easier to just use the images in the slide. Um, so what's going on with CRISPR is a complex, is a system that is capable of taking a DNA sequence that we give it, any DNA sequence that we want, and give it. And it is capable of going into a cell and basically cutting the DNA at a relatively specific location and inserting the DNA that we gave. So that makes this very effective at being able to attach DNA from organism number one, and then putting it into organism number two at a specific location in the genome, at a relatively specific location in the genome. It's not perfect at targeting exactly the right spot we want, which is part of the reason why we don't necessarily do it in embryos, at least in people. But it is something that is specific enough that we can target pretty closely to where we want. Excuse me. So we have a question here. So a somewhat similar process to the cloning vector. The intention is the same. The intention is to take a DNA sequence that we want and put it into the organism. The difference is cloning vectors can be easily moved into bacteria, which will then divide and multiply that vector. They don't work so well in eukaryotic cells, especially in multicellular eukaryotic organisms. Vectors don't usually get replicated in eukaryotes the same way that they get replicated in bacteria, which is why it's important to insert that DNA into an actual chromosome. That's why, and that also goes for our TI plasmid, to get a little bit off topic. Our TI plasmid is so important for plants because it inserts that DNA into the chromosome. The vector that we described previously does not do that insertion that we need. The CRISPR system, CRISPR-Cas9, will do that insertion that we need. So this is why this is better than our bacterial vector if we're trying to insert this into a eukaryotic organism. So let's go ahead and go through some of the different parts and how this works. So first here, we have our CR-RNA. Our CR-RNA is, as you might guess, an RNA strand where we have this protospacer. Now, our protospacer needs to be a complement of our DNA sequence where we want the new DNA strand, the foreign DNA strand, to be inserted. So that's going to be our protospacer, and that's part of our CR-RNA. The other part of the CR-RNA is this CRISPR repeat, this black portion of the CRISPR strand, of the CR-RNA strand. What does the CRISPR repeat do? The CRISPR repeat hybridizes or is the complement to our TRACR RNA strand. So that's this red portion right here. So it is going to base pair with our TRACR RNA strand so that these will now be linked together. Why do we want these to be linked together? And once they're linked together, they are our gRNA. So this is our gRNA complex that we have right here. So why do these need to be linked together? So Cas9 will actually bind to our TRACR RNA. So it will kind of hold on to this red portion as a handle. And once it has this red portion as a handle, it can actually hold onto our cRNA using that TRACR RNA. And then we have this Cas9. Cas9 is the protein with this gRNA complex. Then what we're going to do is we're going to attach to this Cas9 is we're going to attach the DNA sequence that we want to insert into this new genome, this genome that we want to have this DNA sequence in. And our cRNA is going to take us to more or less the specific sequence that we want in the DNA strand. It's going to help Cas9 bind to that DNA strand. And then what Cas9 is going to do is it's going to look for a NGG sequence. N meaning that it could be any nucleotide, but we still need a nearby guanine next to each other, and it's going to cut at that sequence. There are other possible motifs, but this is the one that we're focusing on for this course, so you should keep your eyes open for that in case it comes up. It's going to cut at that location, and it's going to insert the DNA sequence that we want to insert into that location. And that's how the CRISPR-Cas9 works. So it uses the cRNA, I'm just going to say the gRNA, which includes both of these different RNAs. It uses that as kind of a tracking system for the location in the genome that we want it to go. And this can be used to either insert DNA that we want, but also it could be used to insert a just nonsense disruptive sequence to actually break this gene. If this is a gene that's doing something bad, if it's a gain of function gene that's doing something bad, we can actually break it by just inserting a string of various stop codons to make sure that that it doesn't get put to use. So there are multiple possible applications for this. Any questions on this? So if there are no questions on this, I'm going to go ahead and stop there. Is NGG natural? All it takes is two guanines right next to each other. Oh gosh, I'm getting all sorts of. Let me shut this thing up. There we go. Okay. So is NGG natural? It's just two guanines right next to each other. It's going to happen all over the place. This is going to be a very regular thing. If you take any two base pairs right next to each other, that's what, a 1/4 times 1/4, so 1/16. And since guanines and cytosines, I think, tend to be more common than adenine and thymine, it's probably more than 1 every 16 two base pairs kind of thing. So why don't I go ahead and stop the recording for, if I'm going to answer questions not related to CRISPR, why don't I stop the recording for CRISPR?